Okay, for question 2, a student investigated the effect of different growth media on the germination and growth of pollen. So it's interesting, one growth medium, many growth media. Okay, so there are 2 things to be examined here. One is germination, and the other one is growth. So both are different things, right? Germination means from a pollen tube starts to form. So that's called germination. So when it starts to form, it's called germination. Whereas growth means how fast this pollen tube grow. Okay, so there are 2 things to be examined here. So the growth media tested are shown in table 2.1. So you can see that there are medium A, medium B, and medium C. And these are the component in the medium. Okay, so this is the concentration of the components in the medium. So medium A only has sucrose and nothing else. Okay, medium B has got 2 additional chemicals, which are boric acid and calcium. And medium C, have got 2 more components, which is magnesium sulfate and potassium nitrate. So this setup is very important to let us know that, you know, every time when you design an experiment, right, between medium A, B, and C, these are the independent variable. So any differences in the result that you can see will be due to the difference in the independent variable. So that means to say that whatever that happens in medium B, which is different from A, is because of these 2 additional things that are there. So you can compare between A and B. The result in B that's different from A is because of boric acid and calcium nitrate. And then you can also compare between B and C to say that any differences in the result can be due to the presence of magnesium sulfate and potassium nitrate. Okay, whereas for medium A alone, can we say that the germination is due to sucrose? You cannot do that unless you've got another medium which is a control when there's no sucrose. Then between no sucrose and the one with sucrose, any differences in the result is due to the presence of sucrose, and then you can do the conclusion. So I want you to understand the setup of experiment. Any differences in the independent variable, that difference is the one that could cause the difference in the result. Okay, so that's the setup for this one. So figure 2.1 shows the apparatus used by the student in this investigation. So we have a cover petri dish, pollen mixed with a growth medium on the glass slide, placed on top of water to maintain the humidity so that the pollen grain will not dry off. Okay. Here, sorry, where is it? A part 1. Describe how the student could have measured the length of the pollen tubes using a microscope. So here what I want to do is that actually I want to show you the pollen. Okay, what is pollen? What is enter? How do you transfer the pollen from the enter? What is germination? Okay, so enter with pollen. So these are the images to show you. So this is the enter, see the enter of the flower, okay, the male reproductive organ of the plant. Okay, so when the enter is mature, it will dry off and it will split to release the pollen that has been produced within the pollen sac of the enter. Yep. So this one is showing you that you can see a lot of pollen on the surface of the enter because the enter already split open. Okay. So what I want to see here is that originally when it is not mature yet, this is how the enter looks like with 4 pollen sacs, and the pollen is not released from the enter unless and until the enter is mature enough, it will dry off at this line here. So it will split open and then the pollen is able to be released. So when you want to do experiment by transferring the pollen onto the glass slide, you don't use your hand, you don't use a what's that called? Hang on, forceps. You don't use a forceps, okay? You may crush it. So you use a painting brush to gently touch the surface of the enter. So when you touches the surface of the enter, right, you will be, the pollen will stick to the brush. And then from there, you will touch the brush onto the glass slide. So the pollen will be transferred to the glass slide. Okay, so that's how we do it to transfer the pollen. So that's the pollen. And then when the pollen germinate, okay, let's look at pollen tube. Right. So when the pollen germinate, there's a very good diagram here. Okay, so this is pollen germination to demonstrate the growth of pollen tube. So this is pollen. When it germinate means it says that the pollen tube has developed. Okay, now there are two things in the plant that can germinate. You make sure that you know the differences between these two. Do you know what are the two things can germinate? Apart from pollen, what else in the plant that can germinate? You want to try? The seed can germinate. Huh? The seed also can germinate, but the seed is not the pollen. So you need to know the differences between germination of pollen versus germination of the seed, where the new plant will grow. Whereas pollen is not a new plant. Pollen is to transfer the two male gametes to the ovule. All right, the ovule for double fertilization. Okay, so when you say germination means pollen tube is formed. When you want to measure the growth rate, that means you're measuring the length of the pollen tube here. So you can use the eyepiece graticule and adjust according to the orientation of the pollen tube in order to measure its length at different timing of the experiment. Okay, so this is a very good one here. Can you see here? You can measure the length of this pollen tube and the length of this pollen tube as well. So this is where 10 pollen tube, 10 pollen with the longest pollen tube will be selected for measurement in the experiment here. Right, so they look like actually sperms. Okay, they look like sperm. So this one is no germination yet. Okay. A lot of these are not germinated yet. You can see this one got germination. Slight germination. Okay, so that's for overview of germination and growth. Okay, A part 1, describe how the student could have measured the length of the pollen tube using a microscope. Total mark is 3 marks. So first you need to mention what is the magnification that you're going to use to measure it. So for a light microscope in the lab, the magnification that you have learned is either low power, right? Low power that you use is only 10 times 10, okay? Or the high power that you use is actually 10 times 40. So therefore, 1 mark is given for students who say that view the pollen using the microscope at the magnification of 100 or 400. Okay, so there's 1 mark for that. So that means, can you see that when you're asked to describe how, okay, the keyword here is describe how, that means you have to give all the details so the person can follow your instruction and get the results. Okay, so it must be very detailed. So then what you do is that you use the eyepiece. This is what you do, right? Eyepiece graticule to measure the length of the pollen tube. Right, so that's what you're going to do. So measure the length of the pollen tube, and therefore the unit after you measure it could be eyepiece graticule unit, EGU. Okay, so it's in eyepiece graticule unit. So after that, you need to convert the eyepiece graticule unit to millimeter as well as to micrometer. So convert the eyepiece graticule unit to millimeter and then micrometer. So in order to convert, you have to actually calibrate the eyepiece graticule with a stage micrometer. Okay, so once you calibrate eyepiece graticule with stage micrometer, then you can convert whatever eyepiece graticule unit in arbitrary units into a SI unit, which is mm and micrometer. So this is where the practical technique that you learn in semester 1 can be tested in paper 5. There are some students who actually are unable to answer this question. Probably they have forgotten about this micrometry. So make sure you revise everything that you have learned about practical from semester 1 up to now. Okay. Part 2 suggests why the percentage germination was calculated. For this answer, actually, I'm looking for an answer that the students can tell me. Not only you calculate percentage so that you can compare the results, because that one I actually don't understand. Okay. If you look at the passage, in the experiment, we started with different number of pollens on different medium, because it depends how much pollen grains has been transferred from the enter to the glass slide using the brush. And because they're very tiny, we cannot count individually to make sure that for all the medium, we start with the same number of pollen grains. So therefore, for all the medium, we start with a different number of pollen grains, and because of that, there'll be different number of pollen grains that germinate. So in order to be able to compare among the 3 medium, we have to convert the percentage germination in the germination into percentage germination, so that we can compare between the 3 mediums, regardless of the number of pollen grains that we start off with. So that's the answer I'm looking for. So that is the crystal clear answer, that you're telling me you're converting it because you start off with a different number of pollen grains for each medium. That's the best answer. So I'm looking for to allow comparison to be made among the 3 medium, because because each medium starts with a different number of pollen grains. So that's the answer I'm looking for. Okay, so if a student say to allow comparison, I also give mark, but I actually do not know whether the student understand what he's writing, why he need to compare. So this is the best answer I'm looking for, crystal clear answer. You're comparing between the 3 medium, and then the starting number of pollen grains are different, so you have to convert to percentage. So that's what I'm looking for. So if the examiner is strict, if the answer is just to compare, maybe in another exam, there will not be any marks given. So it all depends. So please give crystal clear answer, okay? B, the student carry out t-test on the results to compare the lengths of the pollen tubes when grown in different media. To carry out a t-test, the number in the sample must be counted. So this is actually n, okay? And it is also necessary to carry out 2 calculations from the data. So in calculating the t value, the formula that you need. So this is a formula for t-test. even though you don't have to memorize the whole thing, but you also need to know what are the components you need to make the calculation. So there are three variables here. First one is the mean value for the two samples that you're trying to compare. Okay. And then you've got the standard deviation for each of them and number of samples. So therefore, the question says that you need the number of samples. Other than that, the two other variables that you need is actually the mean as well as the standard deviation. So I'm looking for these two answers to score 1 mark. Anything missing? No mark. Okay. Okay. So that's for part 1. Part 2, state why the student decided to use 18 degrees of freedom in each of the t-test calculation. So you know that for t-test, chi-square test, you need to calculate a standard deviation. So standard deviation for t-test is, because you're comparing two samples, each one of them got their own number of samples. So it's n minus 1, okay, plus n2 minus 1. So it's n1 minus 1 and n2 minus 1. Not, okay, plus. So therefore it's equals to n1 plus n2 minus 2. So that is the standard deviation for, standard deviation, sorry. That is the number of degrees of freedom, okay, for t-test. It's surprisingly half of the total papers that I marked, okay? The students are telling me that the degrees of freedom is n1 plus n2 plus n3 minus 3, I think. So that means the student actually do not understand that t-test is used to compare the differences of the mean of two experiments or two samples only. Okay, so they go and compare the three. So that means not familiar with t-test. So make sure that you're familiar with everything that's in your syllabus. Okay. So the students carry out a number of t-tests to find out if after 70 minutes, the difference in mean pollen tube length is significant. So t value has been calculated here. And this t value, can you see that it's actually comparing the mean between two mediums. So medium A and B, A and C, B and C. So there's a table given here, which is very good. So table of degrees of freedom. So we already calculated the degrees of freedom is 18, because total number of samples calculated is 10, right? They choose, for every time that they use when they measure the growth, they choose the 10 pollen with the longest pollen tube to measure. So the sample is actually 10 for every time, all right? So for this one, they choose the mean value at when the timing is 72, I think. Okay, 72. Am I correct? 70. Okay, so at 70 minutes, right? So 10 samples are being measured. So degrees of freedom is 10 plus 10 minus 2 is 18. So, and in this table, it's interesting that this is a table to show you the critical t value, because they give the probability at 0.05. Probability at 0.05 means probability at 5%. This is telling you that this value is the critical t value, okay, at a probability of 0.05. So whereas if you look at probability 0.01, that means they're giving you the critical t value at the probability of 1%. Okay, so the one that we officially will use for biology is actually the rejection zone is the N 5%. So this is our critical t value. So you take the critical t value and you compare with these three values to make your conclusion here. Okay, this three value. So the next question is asking you to make conclusion. Okay, the next question here. State what conclusions can be drawn about the significance of the differences in mean lengths from the three values of t given above. So what happened is that state what conclusions can be drawn. So I've got some students who directly write the conclusion without describing how the conclusion is being derived. Okay, so here you see what happened is that when three t values are given here, until you want to make the conclusions. So how do you derive to that conclusion? You need to describe fully. Okay, because the critical t value is given, the t value is given. So how are you going to make conclusion based on these two values that are given? You need to describe. So that's where some students did not describe. Okay, and because of that, I think the student cannot score full marks lah. Okay, so first mark is given for student who identify that the critical t value is 2.1. Hello, where are you? 2.1 here. Okay, so if you circle 2.1 here, also you'll get mark, or you describe in words, also get mark. So critical t value is 2.1. So therefore, then you describe that for these two, because the t value is bigger than, so this is bigger than critical t value, which you can put as in bracket 2.10. So therefore, what is the conclusion you can make here is that you have got 95% confidence that because your critical t value is bigger than, sorry, the t value is bigger than critical t, that means is located at rejection zone. So rejection means you reject null hypothesis. That means that there is a significant difference in the growth, okay, in the growth of the pollen between medium A and B and medium A and C. Okay, so that is the full conclusion. Now we don't give mark for reject null hypothesis because there is nothing in the passage or question at all that asks you to state null hypothesis. So therefore, you don't have to say or reject null hypothesis because there's no mark given here. Unless you're asked to write, then you can write reject null hypothesis. Okay, so in the same way, you can do a conclusion that for this example, okay, the t value is 1.94, which is actually less than 2.1. So that means to say that the differences in the pollen, growth of pollen between medium B and C is not significant. So that's a statement that you can write there. So you've got 95% confidence that there is no significant difference in the growth of pollen tube between medium B and C. Okay. So what are the marks given for this question is, number one, state the critical value is 2.10. Number two, you state that for the first and second t value is bigger, you state that it's bigger than the critical t value. Okay, or you state that for number three, the t value is less than critical t value. So this is second marking point. Then the third marking point is therefore you do the conclusion, which is this one or this one. Okay, either or. Okay, now there's one more mark given if the student say that, okay, for this value here, right, this t value is actually bigger than 2.88. That means you can make conclusion that there is 99% confidence that the pollen growth between medium A and C is significantly different. Okay, so for that conclusion also you will get 1 mark. Okay, so that's for this one. Next question. This is a quite interesting question. From the results in table 2.2 and the t-tests, the student concluded that calcium has no effect on germination of pollen grains. So no effect on germination, but it increases the growth rate of pollen tubes. So that's for calcium. Then sucrose is needed for pollen tube germination, but has no effect on the growth rate. So both also talk about germination and growth rate. Okay. So they ask you to use a reference to table 2.2, state the evidence that support this conclusion and the evidence that does not support these conclusions. Okay. So total marks, so they state very clearly here, evidence that support and evidence that does not support, total 4 marks. So what happened is when I mark the answer, before I go into the answering the question, I noticed that some students, right, when the statement that they give here, they write something here. Okay, let's say X, 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 X. They write something here. Now, something that they write here is actually the correct point for evidence that support this conclusion. Okay, but the student is writing in not the correct location. So even though the statement is correct, the statement here is not for the evidence that does not support. So therefore it's contradicting. So therefore I do not give mark. So that means the student actually do not know that the statement that he's giving is actually for evidence that does not support, and he say is supporting. So that is actually contradicting. So I cannot give mark even though the statement is correct because it's not written in the correct location. So it's actually contradicting. Either the student is confused or the student didn't see the words properly or what. So therefore I cannot give mark. So that is actually, to me, it's a waste of point lah. Okay, so I have to write in the correct location. Right, so let's look at evidence that support the conclusion about, hang on, we're talking about calcium, right? Okay, calcium has no effect on germination of pollen grains, but increases the growth rate. So can we say that or not? So go to the medium. Go right to the medium to check. Okay. Calcium present in here and here, but not in A. So based on the t-test, it says that the mean growth of pollen between A and B and between A and C are significantly different. Okay, between these two and these two are significantly different. So we can say that the difference in the, so we can say that the difference in the growth of the pollen between medium A and B and medium A and C is significant. So calcium may actually result in the differences in the growth. Okay. They ask you for the evidence, right? So the evidence is, first, you're talking about the presence of calcium in medium B and C. Okay. So from there, you go back to the results and you check the results that, because the t-test actually, the t-test is done based on this one only, at 70 minutes. So we only look at this 70 minutes for mean length. So it actually shows that the mean length of the pollen tube for medium B and C are both longer than medium A. So therefore, they grow better in the medium with calcium. So that's the first marking point. Okay. So the first marking point is the pollen tube grow longer with calcium in medium B and C. So therefore, this is evidence that support the hypothesis or the conclusion that calcium promote the growth. Okay, first one. Then what about germination? So for germination, we will also look at this timing, since the mean length also we look at 70. So the germination, we also look at 70. Okay, so if you look at 70, you can see that for both medium A, B, and C, the percentage germination is almost the same. They're very close to each other. So I'm looking for the statement that With and without the presence of calcium, the percentage germination for all the three mediums are almost the same, almost the same. So therefore, this is the evidence to show that calcium does not affect germination. So this is second marking point. Okay. Now the third one is, what about the effect of the sucrose? Sucrose is only present in medium A, but absent in medium B and C. So without the presence of other chemicals, with the presence of sucrose, there is germination. Okay, it shows that with the presence of sucrose, there is germination. So this is the evidence you can show that when sucrose is present alone, the pollen can germinate. So it may actually shows that sucrose may promote germination. Okay, with the presence of sucrose alone, there is germination. So sucrose may promote germination. May only, I cannot conclude unless I can compare with the control without sucrose. But it says that it may. Okay, so these are all the three evidences to support evidences that support the statement of the conclusion of the student. Now, what about evidences that do not support? Okay, evidence that do not support is, now medium B and C, apart from having calcium, medium C contain two additional things here, which is magnesium sulfate and potassium nitrate. So therefore, in medium C, when you say that there is a growth, that growth can also be due to the presence of this one, because we are comparing between A and C. So when you compare between A and C, any difference in the result is due to the differences in their content. So the original content is this one, they don't have calcium, magnesium, potassium. For medium C, they've got calcium, potassium, and magnesium. So the conclusion I should be able to do here is the differences is not only due to the presence of calcium, it may also be due to the presence of magnesium sulfate and potassium nitrate. So that is 1 mark. Okay, that's 1 mark if you compare between medium A and C. So if you compare between medium A and B, there's another thing you can look at, is because what about boric acid? Right, so when you compare between medium A and B, you say that in B, the germination is significantly higher. Okay, the tube is significantly longer. Then what is the cause? It can be due to boric acid, it can be due to calcium nitrate. So we cannot say that it must be due to calcium because there are two variables there. So it could be boric acid and not calcium nitrate. We do not know. So therefore, this is the evidence that do not support the hypothesis, the conclusion that says calcium is the one that lead to the the growth. It could be boric acid that can cause the pollen tube growth. Okay, so is that clear? Any differences in the result, you must look at the original medium. What are the differences originally? So that differences is the one that can lead to difference in the result. Spell out the whole thing. Okay, now the third evidence is about the sucrose. So the statement says that sucrose may lead to germination, and sucrose also may lead to growth of pollen tube, which we cannot make conclusion because for all the three mediums, we have the same concentration of sucrose. So because of that, we have no control to compare the effect of sucrose. Does it lead to germination? We do not know. Does it lead to pollen growth? Also, we do not know, because there is no control without sucrose for us to compare. So therefore, we cannot. Okay, how do you write your answer? All the sucrose, all the medium contains sucrose. So therefore, we have no control to compare the differences in terms of percentage germination and also the growth of pollen tube. Okay, so the mark scheme, I'll show you the mark scheme for this particular question here to recap. Okay, So that's here. Okay, mark scheme. Sorry, not this one, the last one's here. Right, this one here. Okay, so recap, evidence that support the pollen tubes grow longer or better with calcium in medium B and C versus A. So this one can support. Then the germination is almost the same with and without calcium in medium A, B, and C. And then thirdly, the pollen germination occurs with sucrose alone. So with sucrose alone, the pollen can germinate. So it could be sucrose can promote germination. Could be, Evidences that does not support. Boric acid could be the one that cause an effect on pollen tubes in medium B as well as medium C. Okay, medium C also contain magnesium, potassium sulfate, so they might be the one that causes the pollen tube growth. Last one, all medium also have sucrose, so there's no way of knowing the effect of sucrose, whether it will cause germination, it will cause elongation of the pollen tube. Okay. One more thing I want to mention is that when you do an experiment and do a conclusion, in the conclusion, you cannot say the result proved that. Okay, the result never proved anything. Okay, the result cannot prove. The result only shows something. Okay, or there is a 95% confidence that this is higher than that. Okay, so this may cause an increase in the growth rate. So never use the word the evidence or the result proves something. Okay, we cannot prove something. We can only show something. Okay, so conclusion, show something, not prove something. Okay, so I think that's all for today for the discussion.